Hello viewers, today we will discuss the different types of receptors and how the receptor propagate the signals, that is the transduction mechanisms of the receptors, how the receptor produce the effects, and our today's learning objectives that by the end of this session you will be able to understand and discuss the different types of receptor that major four types of receptors that we will discuss, and then we will discuss the signaling mechanism of the receptors, that is the transduction mechanism of the receptor, how the receptor transmit or propagate the signals after binding with the agonist, and then we will discuss the impact of signaling mechanism on the pharmacological effects, that how the different type of receptors propagate the signals through the signaling mechanism or the transduction mechanism, and what could be the resultant pharmacological effects. So these are the four types of receptors. This is serpentine type of receptor, or we call it as the G protein coupled receptors. This is the cell membrane. And this is the another type of receptor, that is the ligand gated ion channels, and these are the enzyme linked receptors, and these are the intracellular receptors. So we will discuss these four very important families of the receptors. Now this is the serpentine type of receptor. Why we call it serpentine? That because it has 7 transmembrane helicals. This is the outer domain, but the binding domain of the receptor. As we will talk about general function and the general structure of the receptors, we have talked about in previous presentations that the receptor have 3 domains, that is the outer domain, binding domain, transmembrane domain, and internal domain. So this is the outer domain, and this outer domain is for the binding of the receptor, and this is the transmembrane domain, and it has got 7 transmembrane helicals, and it is linked, internal domain is linked with the G protein. And because this is the characteristic of the serpentine type of receptor, as the receptor has 7 transmembrane helicals, that's why we call it as the serpentine receptor, and it is linked with the G protein. Now at this level, no agonist, no drug come and bind. Receptor is in a very normal resting state, and this is linked with the G protein, and G protein is inactivated, so it is bind with the guanosine diphosphate GDP. When the drug come and bind with this receptor, now this transmembrane domain, they shows the intrinsic activity. And what is the intrinsic activity? That it activates this G protein, which is linked with the internal domain of this serpentine type of receptor, and now the GDP is converted into guanosine triphosphate, that is the GTP. Now this is the intrinsic activity. These when the drug come and bind with this serpentine type of receptor, it changes the arrangement of the 7 helicals, and in response to this intrinsic activity, it activates this G protein. Activates the G protein, it means now the activated G protein is linked with the guanosine triphosphate, and this activated G protein activates the effector protein. That effector protein is now be able to produce the effects. Now in this situation, this G protein which is being activated, it activates the another effector protein, and this another effector protein will produce the effect through secondary messenger system. That This G protein, there are three types of G proteins. That is GS, S for the stimulatory, and this stimulatory GS stimulates the adenylate cyclase enzyme, and this adenylate cyclase enzyme increases the formation of cyclic AMP, that is the secondary messenger system. And now it is the duty of that cyclic AMP to produce the effects. Another G protein is GI, that is inhibitory. When it is activated, it inactivates the adenylate cyclase enzyme and decreases the availability of the cyclic AMP. And this cyclic AMP is actually regulates the phosphorylation of the proteins. And the third type of G protein is the GQ, which activates the phospholipase C. And this phospholipase C finally activates the IP3 inositol triphosphate and diacylglycerol DAG. So this secondary messenger, one opens the calcium channels and another mobilizes the calcium inside the cell. So this generally increases the mobility of the calcium inside the cell and produces the effect. So these are three types of G protein, and the resultant effects of the drug depends on this attached G protein with the serpentine type of receptor. If the serpentine type of receptor is linked with GS, it increases the adenylate cyclase enzyme and increases the cyclic AMP, and then it is the duty of cyclic AMP, which is the secondary messenger, to produce the effect. If this same serpentine type of receptor is linked with GI, and by binding with the agonist, it activates the GI. So this GI means inhibitory. So it inhibits the adenylate cyclase enzyme, and because of the inhibition of adenylate cyclase enzyme, the cyclic AMP is deficient, and all the effects are produced because of the deficiency of the cyclic AMP. And likewise, if the receptor is linked with the GQ and it activates the phospholipase C and it increases the formation of the IP3 and DAG, it mobilizes the calcium, so it produces the effect. So the important example of that is the alpha-1 receptors. They are the serpentine type of receptor. Last time I have told you that the majority of the drugs, they are acting through serpentine type of receptor. So alpha-1 receptor, they are linked with the GQ. So when they are activated, they increases the availability of IP3 and DAG, and it mobilizes the calcium. So this alpha-1 receptor's location is the smooth muscles of the blood vessels. So when there is increased mobilization of the calcium inside the smooth muscles of the blood vessels, it causes the vasoconstriction. Likewise, GS is linked with the beta receptors, all the beta receptors, beta 1, 2, 3, 4, and 5. All these beta receptors, they are linked with GS. So when they are activated, they stimulates the adenylate cyclase enzyme and increases the availability of the cyclic AMP and causes or regulates the phosphorylation of the proteins and then produces the resultant effects. Another example is alpha-2 receptor. It is linked with GI, and these alpha-2 receptors, they are autoregulatory and they are present presynaptically, and they auto-regulate the release of the norepinephrine from the synaptic nerve, and it regulates the release of the norepinephrine, that is the neurotransmitter, at the synaptic level. So all this about this G protein, and what is the conclusion? Conclusion is that is the resultant effect of the drug depends on the G protein, which is linked with the serpentine type of receptor. Now, what is the activation of this G protein? Now, this is the situation, this is the serpentine type of receptor. This is the G protein. G protein has 3 subunits, alpha, beta, and gamma. And this receptor is in a resting condition. No drug comes and binds. It is unoccupied. So this G protein is inactivated and bind and attached with the guanosine diphosphate. And this is the effector protein, that is the adenylate cyclase enzyme, which when activated, it increases the synthesis of the cyclic acid. When the drug comes and bind with this receptor, it will activate this G protein. This is actually the intrinsic activity, that when the drug comes and bind with the receptor, it attracts the G protein towards itself, and the inactivated G protein, which was attached with the guanosine diphosphate, is now attached with the guanosine triphosphate. Now the receptor is activated. It shows the intrinsic activity. So definitely the receptor will produce the effect. And how it produces the effect, that this alpha subunit, which is linked with or attached with this guanosine triphosphate, it detaches from this receptor and it activates the effector protein, that is the adenylate cyclase enzyme. And this enzyme will produce the effects. When this drug leaves the receptor is now free, so this G protein again goes back to its original normal position. That is, again this the alpha subunit is going back and is attached with the beta and gamma subunit, and the guanosine triphosphate is now again converted into the guanosine diphosphate, and again this resting situation comes back. So this is the situation of the G protein, whether it is GS, whether it is GI, whether it is GQ, but all the resultant effects of the drugs which come and bind with the serpentine type of receptors, they produces the effects, and their effects depends on the type of G protein which is attached with the serpentine type of receptor. Another family of the receptor is the ligand-gated ion channels. And these receptors, they are responsible for the regulation of the flow of ions across the cell membrane. You see, the domains are same. In the previous receptor, domains are same. In this receptor, domains are same. That is, this is the cell membrane, and this is the outer domain of the receptor, or the binding domain of the receptor. This is transmembrane domain of the receptor, and this is the internal domain of the receptor. And the activity of these channels is regulated by the binding of the ligand to the channel. Now, in this situation, or in this type of receptor, the signaling mechanism is totally different. No G protein is linked with this receptor. It has totally different signaling mechanism. That this is the agonist when come and bind with this alpha subunit of this receptor, and this receptor is linked with the ion channel. So, if it is linked with the sodium channel, so what happens when this agonist come and bind with the receptor? It shows the intrinsic activity. And what is the intrinsic activity in this receptor? That this subunit will contract in this direction, and this subunit will contract in an opposite direction. So when both these subunits, when they contract in opposite direction, so there will be the opening of the channel. And when this receptor is linked with the sodium channel, there will be the entry of the sodium inside, and it causes it generates the action potential and produces the excitatory response or excitatory effects. And the important example of such sort of a receptor is the acetylcholine. That acetylcholine is the ligand for this ion channel receptor. Ion channel receptor is being activated by the attachment of the agonist, that is the acetylcholine, and it creates the action potential due to the influx of the calcium. If the same receptor is linked with the potassium channel, so again when the drug comes and bind with the same binding domain, and it shows the intrinsic activity that this subunit will contract in this direction and this subunit will contract in opposite direction, so there will be the opening of the potassium channels, so it causes the hyperpolarization and it produces the inhibitory signal. So this is actually the channel. It could be the sodium channel, it could be the potassium channel. So this is the channel and this is the binding subunit of the receptor, and this is the binding domain of the receptor. When the drug comes and bind with this binding domain of the receptor, now this subunit will contract in this direction and this subunit will contract in an opposite direction. So there will be the opening of this channel and there will be the entry of the ion, whether if it is sodium, so there will be the entry, and if it is suppose is linked with the potassium, and again the agonists come and bind, again it contract in an opposite direction and this is in another direction, so there will be the exit of potassium from the cell, so it causes the inhibitory effect. So this is about the signals, that these are the signals produced by the entry of sodium, and these are the inhibitory signals which are produced by the exit of the potassium, and we call them as the excitatory post-synaptic potentials or inhibitory post-synaptic potential. Excitatory post-synaptic potential, because when there is the activation of the sodium, it causes the depolarization, so it produces the excitatory post-synaptic potentials. When there is the mobilization of the potassium, so it causes the hyperpolarization and it creates the inhibitory post-synaptic potential. So this is all the resultant effects of the drug. They depends on the type of channel to which the ligand-gated ion channel receptor is being linked. If it is linked with the sodium, the effects are totally different with if the receptor is linked with the potassium channel. So this is another very important and a very common type of receptor to which the majority of the drugs, they come and bind and produces the effects. Now the another type of receptor is enzyme linked receptor. An example of this enzyme linked receptor, again you see the domains are same. This is the cell membrane, this is the outer domain which is the binding domain for the agonist, this is the transmembrane domain, and here the transmembrane domain or the internal domain of this receptor is linked with the enzyme, that is the tyrosine kinase enzyme. That's why we call such sort of receptors as the enzyme linked receptor. And the most important example is the insulin. When the insulin comes and bind with this receptor, it activates this tyrosine kinase enzyme, and this tyrosine kinase enzyme causes the phosphorylation of this tyrosine residue of the protein. So the main function of this receptor is the autophosphorylation and the phosphorylation of the tyrosine residue, which generates the proteins in response to this phosphorylation. So what happens when this receptor is being activated? This is the cell membrane, this is the outer domain of the cell receptor, the binding domain of the receptor, this is transmembrane domain of the receptor which shows the intrinsic activity, and this is the internal domain of the receptor. Now when the drug, like we are getting the example of the insulin, so when the drug comes and bind with this binding domain of the receptor, then what happens that the receptor causes the autophosphorylation, that it is phosphate bonds they are being attached with the receptor. So this is called the autophosphorylation of the receptor itself. and it causes the phosphorylation of the protein. And in case of insulin, the protein is the glucose transport protein. And the function of this glucose transport protein, which was being generated at the cell membrane level. So what happens, and it's very clear from the name, that it transports protein for the glucose. So it transports the glucose from outside to inside the cell membrane, and it causes the utilization of the glucose, and it causes the hypoglycemia. And this is the very important type, but here you can see that the signaling mechanism is totally different from the serpentine type of receptor, that is G protein coupled receptor, and the ligand gated ion channel receptors. Here the signaling mechanism is totally different, which is responsible for the resultant effect, that is the generation of the glucose transport proteins, and these proteins, they increases the transport of glucose from outside to inside. Now the another receptor is intracellular receptor. Now here there are a few exceptions always, but more or less the story is same. What's the difference in this type of receptor? That this receptor is present inside the cell. This is the cell membrane, and this is the cytosol, and this receptor is present in the cytosol. And this example of such sort of intracellular receptors is the steroidal hormones. All the steroidal hormones have a receptor inside the cell in the cytosol. And these steroid hormones, they are highly lipid soluble. So because of their lipid solubility, this hormone diffuses through the membrane very easily. And this hormone, it binds with the receptor which is present in the cytoplasm, and it forms the drug receptor complexes, or the hormone receptor complex. Now you can see over there, what is the difference between this unoccupied receptor and this drug receptor or the hormone receptor complex? You can see the change in this receptor structure. This is called the intrinsic activity. This is called the intrinsic activity. Now the activated ligand receptor complex enters the nucleus, and it migrates to the nucleus. It binds to the specific DNA sequence. We call it as the response element, which has the same structure. So it's exactly like a lock and key, that this drug receptor complex exactly fits on this sequence, that is the response element on the DNA. When it fits on the response element of the DNA, it causes the transcription, gene transcription. It triggers the gene transcription. And this transcripted messenger RNA is translated into the protein that alter the cell activity. Majority of this hormonal type of drugs, steroidal hormones type of drugs, and they increases the generation of the protein. They have the receptors inside the cell. Now, what is the difference between the other receptors that we have discussed? Difference is that in those receptors, they have the binding domain outside the cell membrane. Here the domains are same, but the receptor is not present outside the cell membrane. It is present inside the cell in the cytosol, but it has the binding domain. It shows the intrinsic activity, and the intrinsic activity of this drug receptor complex or the hormone receptor complex is that it transports this complex inside the nucleus and bind with the DNA, and finally is responsible for the synthesis of the protein. So all we have done about the four very major important type of receptors. We have done the signaling mechanism, how this G protein coupled serpentine type of receptor, they are producing the effects, how the ligand get it, ion channel receptor produces the effect, and how the enzyme linked and intracellular receptor producing the effect. And they have totally different effects, and their effects, they are depending on signaling mechanism. Thank you very much.